So now our next in that part is topic called microscopic count. Here we are. Do you know about the Newbar's chamber? Or anyone has used this hematocytometer or Newbar's chamber? Newbar's chamber. Have you done the total cell count? Total cell count. How you have done? So it is based on the one. Here you can see one slide is there. Location of the square here into in between. So we can use Newbar's chamber. I mean, just one thick slide. It's the special slides are there. Generally for yeast count, or sometimes for the E. coli, or sometimes we may go for the WBC and RBC. For that, we may use this kind of a chambers. So it became very easy. Directly, square wise, within one square, how many cells are there? We can count, and accordingly, it will be multiplied. There will be a multiplication factor given to it. That within one area, if I'm looking that on one 4 boxes are there, within the one box I'm finding 8, somewhere it's 7. So it is like a 12 box I'm reading. How many it is there? So average, some numbers are coming, we can multiply. So it is average certain numbers are coming. So it is the very easy and convenient method for microscopic count for the cell. Yes. There is a 5 to 6 minute long one documentary is there. So let's see about it. Yes,
A
this one is a Newbar's
chemocytometer
chamber.
is a device that is used for
Cytometer or
counting
a Newbar's
cells.
chamber.
It's a modified microscope slide containing 2 identical wells or
There are the two
chambers
sectors
into
are there.
which a
One at
small
time
volume
can
of
have
cell
the
suspension is
different
pipetted.
things to be get
We have
analyzed.
already removed 100 microliters of our cell suspension and placed it in a microcentrifuge tube. Dilute the suspension by adding 100 microliters of trypan blue.
Generally for
Trypan
the
blue is a
white blood
dye
and
that
others,
helps
may
us
go for the
distinguish
different
between
types.
living and dead
It
cells.
may get
The dye
the white
passes
blood
through the
cells
membranes
generally
of dead
not
cells,
having
so
any
they
color.
will appear blue
Color
under
code
a
is
microscope. Living cells exclude and will appear mostly clear. Load both chambers by pipetting the suspension under the cover slip.
So below is a chamber and upper one is a one cover slip. So as you are moving by capillary action, everything will start to move. It's called the chamber recharge.
Now place the hemocytometer under the microscope. Each
I think
chamber
it's
is
not
divided
9.
into a
Yeah,
grid
this one is
pattern
like a
consisting of
sectors.
9 large squares.
9 blocks are there.
Each square has the same dimensions
Here
and
it is having
contains
the more
10 to the
grids
negative
comparatively
4th power
others. Here you can see the
milliliters
some
of
cells.
suspension. The rules for counting cells sometimes differ from lab to lab. In our lab, we count cells in the 4 large corner squares and the center square. Let's count the cells in the first square. 1, 2, 3,
એમાં એ રીતે
4,
ડ્રો કરેલી છે
5,
ક્લાસ
6,
સાઈડ ઉપર લીટી છે
7,
એટલે તમને
8.
બોક્સ જ સીધા દેખાય.
So what about the cells that are touching
slide
the
by-
outside
slide by
boundaries
level.
of the square? In our lab, we count the cells that touch the top and left boundaries, and we ignore the cells that touch the right and bottom boundaries. 9, 10. We need to count the number of both living and dead cells. Remember, the dead
So
cells
in one
will
box,
appear
how
blue.
many cells are there? We can find. So it
Occasionally,
is the
you will
color
see
wise.
artifacts,
If it is a blue,
objects, or
that is
debris
a
that
non-viable
appear
and
blurry
viable.
and
That is a
don't
dead
have
cell.
a well-defined shape.
Viability can
This
be
is an
confirmed
example
based
of
on
an
our
artifact.
color code.
We won't include it in our count. Proper storage, cleaning, and
Artifact
handling
means
of the
it is a
hemocytometer
dust.
will minimize the number of artifacts.
Sometimes we may
There are
consider or
10
not. So
viable
there is no
cells
color,
and
but
1
something is appearing.
non-viable
So it is
cell
called
in the
artifact.
first square. Now,
In reality,
the
there
top
is
right
nothing.
square. There
And
are
yes,
9 viable
here we are
cells
looking with the
and
L
no
shape,
non-viable cells.
If it is
Next,
coming in
let's
here and
count
other
the
borders,
bottom right
where
square.
to count one step. So it is otherwise get misguided. Sometimes we may count
There
in the
are
upper box, lower
11
box.
viable
So it is
cells and
advisable
no
to go for an
non-viable
L
cells.
state.
And now the bottom left square. There are 10 viable cells and 2 non-viable cells. And finally, let's count the cells
Blue
in the
is the
center
dead,
square.
non-viable.
Sometimes cells will appear as clumps or small groups.
That
It
is
may
what
be
I'm telling.
difficult to
We are looking
determine
for the L
exactly
state.
how many
Here
cells
is a
are in a
box.
group.
So here
The
we
method
go
of
in
counting
L.
clumps of cells
Next
differs
time,
from lab to lab,
for the
so
bottom
be
of
sure
the
to
cell,
follow the
it will be
procedure in your lab.
So we will not
We
considering
will
in here.
count this clump as
It will
2
be there.
cells.
So it's, if
There
it
are
is someone is a
14
top
viable
over
cells
here, so we are
and
not going
no
to count over.
non-viable cells in the
So it is
center
the
square.
average. We are going to consider this an average.
The total number of viable
Maybe
or
one or
living
two
cells
maybe
from
get
all
mixed,
5 squares
maybe.
is 54. The total number of non-viable cells is 3. Now that we have
No, we
counted
are going
our
to count
cells,
each.
there are several calculations
If it
we
is
need
a
to
9 box
perform.
are there, so which box
First,
we are going to
let's calculate the
over
percentage
it, but
of
it in a
viable
format of
cells.
L.
Here's the formula. 54 viable cells divided by 57, the
So how we
total
can
number
count the
of cells,
percent?
gives
Total
us
number of cells divided by viable
0.947.
cell divided by total number of cells
Multiply
multiplied
by
100.
100,
It's going to give a
and the percentage of viable cells is
Present and absent. If I'm going to look the
94.7%.
percentage wise.
Next,
So today how many?
let's
if I'm looking at
determine the
24.
average number of
Strength is
cells per
50.
square.
So
We
24 divided
counted
by 50
54
multiplied by
viable
100.
cells.
So to the strength.
We divide 54 by
Like,
5
it's the
because
viable
we
cells,
counted in
percentage
5
of viable
squares.
cells.
The average number of cells per square is 10.8 cells. Now let's calculate the dilution factor. The dilution equals the final volume divided by the
It is
volume
the
of
dilution
cells.
factor also. That
Our final
sometimes
volume is
here it is not
200
required to go into
microliters
detail. Just for
because
you
we
it's a
started with
method, one
100
method we are looking
microliters
to
of
do.
cells
It's enough
and added
microscopic
another
method we are
100
how we are
microliters
going.
of trypan blue.
Sometimes if you have taken the 10 microliter and you have loaded, you find that it is high density number of the cells are there. So you may dilute it. You may dilute 10 times, again load over it. So at least you are able to count the cells. So whatever the number is coming, we have to multiply. So when you are looking like a per ml, how many cells are there? So based on that, we can calculate over it. So it's a dilution factor. And ultimately, multiplied by, if you're looking for the per ml, we're going to multiply it with their 10⁴ factor. So here, just for your information, this formula is not required right now. It may be as one of the practical, maybe there in your practical sessions, maybe here in another stems. Okay, there it may be given for the total cells count for the yeast, or else in immunology. Next semester you may having that next to next. One paper is there in immunology. Okay, yeah, I think in 5th semester. There you are going to have the blood cells. Blood will be taken and you are going to calculate white blood cell and the red blood cell. The red blood cell always there in a higher density. Okay, so it will be counted in between. Here it is here. If you're looking for the grid lines, many grids were there. So in between there is a larger grid is there. So you need to use the optical like it is. You are looking for the 10x. So you're going to increase the power in 100x. So one box will be become large. Then it became easy. Otherwise, within one box you will find thousands of the cells. It's really difficult to calculate. So just one box is again divided into 25 sectors. So each may be going to reduce accordingly. So that may be a practical, maybe been asked or perform in next immunology related paper. Okay, so what are the advantages and disadvantages in this technique? It is very easy and perform, easy to perform. Here we not require any much incubations like in pour plate and other techniques we are looking. On the next day we may going to count. Okay, then we are not looking for the spectrophotometric assay also. It's become very easy and convenient for a process. But sometimes there is some disadvantage that every lab cannot sometimes afford because it's that Newbar's chamber. It's costing about 5,000 rupees. One time is coming, one slide, it's about 5,000 rupees is there. So it may be, but for research work and going on, so 5,000 is nothing. But the chambers that slide is coming about worth 5,000 rupees. And yes, sometimes if the concentration is very high, then we need to go for again and again dilutions. So if the protocol is set, then we can. that already we know that sometimes the density is coming in this sectors, so we may dilute it accordingly. Like 10 microliter, or we are taking the 100 microliter. Okay, rather 100 microliter, we may take only 10 microliter and dilute it. Otherwise, go for the serial dilution first and then go for the loading on a cell, because it is like one, if you are looking, earlier also I have told that a single colony, when you inoculate 1000 organisms, on a next day, on 24 hours, we are getting the millions of the cells. Okay, from that, if you are taking 100 microliter, it's really difficult to observe the cell. So in which growth phase, yeah, using this microscopic observations, it became very much tedious to enumerate or count the cell. So over there, we need to go for the dilutions with certain factors. So serial dilutions, it may become must. Otherwise, you will find the so many bacteria are there. We are looking that in this box, maybe I'm looking for the 7 to 8, but you are finding 200. It's became our limitations, our eyes limitations also there, because there is so many dots we will observe, whether it is I have calculated or not. And within that, sometimes you are reaching at 150. Someone is asking, or someone is looking to take something. Okay, then you may forget where I was. It became really difficult. So it's a number wise there. Then we need to go for the dilutions also. So these were all the direct method. Now we are looking for the indirect method for the microbial growth. There are the three major: turbidity, metabolic activity, and dry weights. One of the most important being used is the turbidity. And on this Thursday, we are going to perform about it. So on a Wednesday, we are going to, in my lectures, we are going to prepare at least N broths, nutrient broth. So when you are coming on a Thursday, those who are residing here in somewhere in especially hostels, so what they can do, they can inoculate our flask. Between the two burners, they will inoculate an hour based. Every hour, we are going to take the some amount of it, about 10 ml and others. Then we are going to measure on optical density at 600 nanometer. Why the 600 is been kept? Here you can see these observations, right? There is the two tubes are there. One is the normal, one is the growth is going on. Okay, when you are going to put over it on a tube, some reading will come. So it is what, maybe you are knowing the Beers and Lember's law for the spectrophotometer. So every time any lights is passing, okay, something is get absorbed, other is transmitted. We are going to measure the transmitted light. Okay, so optical density, we are considering the transmitted light, and it is going to give into certain points, like 0 point or something is coming. So we are going to measure it. So one at a time, we are not going to measure all two things. What we will do, every hours. So on that day, on a Thursday, maybe then after my lectures will be there, right? Second to third, fourth will be there. So there we go for the measurement, or else maybe my first lecture, second or third is there. At least one or two, take the permission from the sir. I think maybe it's Jayanti sir and the others, one or two maybe. Okay, take the permissions between the two burner. Keep the some centrifuge tubes autoclave. So on that day, we are going to autoclave certain tubes also altogether. So you may take about 2 to 3 ml only, not more. 2 ml, you may take the sample, and then after we are going to measure OD together. But that tube you need to keep within the freeze. Otherwise, what's going to happen? That metabolic growth is going on if you are keeping outside. So every hour you are going to take the certain amount and you are going to put into a freeze. So from it may be maximum we are going to 3 to 4 flasks. So all you people may divide into 4 groups, and we are going to prepare about 100 or 150 ml flask. Okay, one in time, one students may inoculate all together all and keep into the incubator at 37 degrees centigrade. And every hour, that 8:30, then 9:30, till we are going to reach at certain limits. Here we will going to do some different parts that we are preparing the nutrient broths. Okay, nutrient broth, whatever the amount is written, generally we are taking or adding like 28 grams. If I'm not wrong, just I have to recall, like 28 grams per 100 ml. Suppose we are adding, or 2.8 gram, sorry, 28, I think in 1 liter it may be. So 2.8 gram in 100 ml, 2.8. So we will take 1/4 of it. What we are looking, that bacteria initially get adapt, going in leg phase, then log phase. Okay, leg to log to stationary phase. So if you are going to give the more nutrients, so they will easily like to do their work. So we will add, if it is 2.8, only 0.7 gram in 100 ml. Okay, so we are going to prepare the 4 flask. Okay, 150 ml. Accordingly, you may calculate, and you are going to prepare it. Along with it, on a Wednesday, I will provide this some blank scientific tube to also you people. We are looking to do a MBRT test, methylene blip. So if and tomorrow on the next day when you are coming from your home with certain milk, about 8 to 10 ml milk you are going to bring. What is our motto over it? That is there any bacteria present or not? So some people, if you are may take 1 or 2 tubes, you may go in a canteen. Okay, so you are going to bring the milk. One is a normal without boil, and another may be boiled. So we hope that bacteria were killed. Okay, we are going to add the methylene blue over it. Okay, so on a next day we can observe them. Okay, so on a Wednesday we will prepare the media, nothing else. That is our target. Okay, media preparations and part is done. Scientific tube, some few at least required for the each group. We are going to also sterilize it. And certain scientific tube I will provide in a group wise. It is not for the all. Otherwise, everyone is bringing and so many things. It's not a. We are just looking for the observing something. So okay, so every group wise maximum we can go for the 4 to 5 tubes. It is not compulsory. Okay, at least 4 tube is there. We can go for it. It is like one is the Amul direct pasteurized or other brand is available. We may go for it. The raw material. Sometimes some are telling that हमारे यहाँ गिर गाय नो आवे છે નોલા ફલાણા ભાઈ નો આવે છે બરાબર છે બીજું તો કાઈ હોય નહીં સમ્રાટ ડેરી નસક ડેરી ને ઓલોન ડેરી એટલે એમાંથી ઘરેથી તમે લઈ આવજો અને we may go for it. So just what we are looking on a next day. Okay, on the next day when you are going to check. So on a Thursday on practical time we are going to inoculate it. On a Friday maybe at 12 o'clock we may directly go for their reading to check it out whether there is any color change is there or not. So you will surprised every time people get surprised at what's happening. Okay, the milk what I have kept over here. Okay, that is some boil will be there. We at least we can trust. Okay, you will say that સમ્રાટ ડેરી દૂધ સારો છે કે નહીં અમૂલ ડેરીનું દૂધ અમૂલનું દૂધ આવે છે કેવું છે બરાબર છે મિલ્ક જ લઈ આવજો બીજું કઈ ન લઈ આવતા છાસ ને આ તે માખણ કેવું આપણે કોઈ નથી જરૂર બરાબર છે only drink. That's why I'm telling. You may autoclave it, you may not. That is not a problem. બરાબર છે કારણ કે આપણે તો જો ઘરે દૂધ આવતું હોય બરાબર છે એમાં કઈ ઓટોક્લેવિંગ હોય છે એટલે સેન્ટિફિટ આપણે વોશ કરીને આપ યુઝ કરશો. We will go for ઓપન ક્યાં કરશો તમે ઘરે બર્નર વચ્ચે તો પછી ગેસ વચ્ચે પોસિબલ ઈ નો થાય કઈ એટલે તમે ટ્યુબ તો ઓપન જ કરવાના છે બારે તો વોશિંગ હતી કે નોન વોશ ઓટોક્લેવ છે એવા એવા નખરા કાઈ નથી કરવા આપણે. આપણે એવું નહીં કરીએ. In this protocol milk is taken raw material. Okay, autoclave tube માં નથી લેવામાં આવતું બરાબર છે એટલે આપણે નવી વસ્તુ નહીં કરી. Okay, so it is based then after we can calculate. So if the transmitter is there we may go for the OD and based on that graph preparations is there. So it became very easy. Turbidometric method is very easy and convenient to perform, right? So yes, another things already I have uploaded journal for you people. So on a Thursday, not before, no one is going to come to my place also. Thursday I will check your record book. Those who are in online and offline, those who are online they can pass to their colleagues to check for the checking it on a Thursday. Okay, maximum Thursday or a Friday or at 12 o'clock we are going to meet, so you can show it. Okay, then after I will give about 4 or 5 days. So today is the 4th. Okay, we are going to check on a Thursday. So I hope it will be get completed before it. Yeah. Record books. Okay. Record book will be completed. And then after, I will give about 3 to 4 days to write it down in a journal to check it out. Yes. Yeah. મંડે જર્નલ ની ડેટ આવશે બેટા 15 થી હું તમારી 15 કે 18 તારીખે તમારી યસ બી રેડી between 15 to 30th October your final practical exams will be there between 15 to 30 October there will be your practical exam so each એકવાર સાંભળી લો please listen first પછી તમે પૂછજો વાંધો નહીં please listen first between 15 to 30th October earlier also I have told again I'm telling between 15 to 30th October your practical exam will be there so you are having only 3 I think that is a microbial physiology biochemistry and chemistry so whichever date will be convenient for their each professors or the group of your teachers will be there they will finalize the date it will be between 15 to 30 so in accordingly your theory exams will be there sorry theory days will be there before 31st we have to complete the syllabus right so maybe if my syllabus is completed not completed I'm going to call you for it on that day if the practical sessions will be there that day with theory teaching hours maybe not so in my case I may require 1 or 2 days right on first day it will be inoculation second day it will be observations so I may set near about on a third week of the October third to maximum fourth because otherwise it will be like complicated that chemistry and physiology and the biochemistry they may say we want to set in this stage so I'm looking to complete in a your early so you will be not in a burden zone from my end at least for the completion of journals so be ready in a third week of your in this October month it's the first week is just going on in third week your exams maybe or it is surely will be there so accordingly on a Thursday to Friday as you are going to linger okay journal writing I have already uploaded and another thing if you can take more time I may fix some certain dates for the journal submissions and I'm telling in advance also okay afterward as you are coming sometimes people will call sir my date is this I haven't submitted yet I will allow always on day before also I will allow but I'm going to put only a marks accordingly I will always allow you are not coming with journal I will allow I just put the 0 marks that is very good faithfulness from my end you can get from you for you people okay I will give justice to all it means those who are early bird they have performed well they submitting in time and those who are coming at the last so what is the justifications for them those who are completed if the 5 marks I have allotted for the journals I can put the 5 marks yeah I will check it is done properly or not something is written something is not there they accordingly I may reduce the marks otherwise I can put the full marks and those who are coming at the last sir I don't know how to download where to write I haven't submitted record book it will go it will not so on an exam day you will require to bring your record book as well as your journal okay if you are coming at the last day and you are submitting your journals you haven't submitted your record books that is a continuous evaluation in practical session is not there so accordingly that part also get reduced so I will give the justice to all those who are coming early it will be rewarded accordingly those who are coming at the last the gradient will be there in my marking system those who are coming at the end or on a, sometimes they will not come with without record book accordingly I reduce the mark in the another sessions what I have kept in the continuous evaluation those who are I know many of the faces was maybe not a name okay but when you are coming for the Viva and others I will prepare my Excel sheet everything before you okay everything will be ready just I need to check and I can put the marks so I will allow you are telling that keep on a Monday on a 10th day or in a month of the 21st you can submit after practical Viva also I never mind yes after the semester also yes also bachelor's Yes, that's you are knowing some. So there will be no harm, but accordingly you will get rewarded from my side. Because our time limitation is there. So today is the fourth. If you are looking for the third, so on a time, I'm not giving that on same day you will come. On Monday, I'm telling you, you may come on the Thursday with your record books, and only two things will going to remain, right? Only two things will going to remain. This is related to growth curve. Okay, growth curve and MBRT test. So on a Wednesday, I will make you to write it down that protocol also. Okay, on a Wednesday, we are going to do that part also. So practical sessions, to write it down record a book, everything will be done before a finalized date, right? So at least you can show on a Thursday, maximum on a Wednesday, then after on a Friday to your record book to me. So our practical sessions, yes, on that day we are going to do few only things, right? Practical part, one person is going to take the just 2 ml or 4 ml amount in a scientific tube. Nothing else is required to do. So we are going to sit idle. Those who are able to do this, those who have completed their record book, I will check and they can start their writing in journal. So within that, as you are going to get signature date as early from my end, you will be going to get more from my end. That is sure. Okay. Already I have told what you are going to write in a journal. If you are going to write anything additional, right, something are additional is there, then you need to show me. Otherwise, you can start writing journals. I will never mind about it. If you are ready, okay, just show me that certain additional part is there that I will not allow other before my signature, because it's sometimes it's not maybe a written or correct is not there. It is very seems clumsy otherwise. So just show me. If you are free, start to write journals. There is no harm into it. Otherwise, if you are looking that, I'm going to, so you can put the some space, 1 or 2 paragraph, right, while writing the journals. Already I have uploaded, it will be about 22 pages. Maximum it is 22 page, and all are maybe a half of page only. Every experiment I have kept starting on the next new page. I can just show it. If just no. Yes, let me show. So here you can see that the whole journals I have uploaded with the page number. Okay. So if you are looking to, so first is related to preparation of a liquid media. So it is written properly. In your, when you have taken the snapshot, that I have written that not introductions you have to take from here. So introductions, it may be not more. Okay, it will be a maximum 1 or 2 page. Yes, instructions to prepare, how to prepare. Okay. It will be there. Protocol I have, this one is a part of introduction. This one is a part of introductions. And here you can read afterwards, not. Requirement, additional note, if any. If you are looking to write it down this additional note, then you have to show. Then observations, already I have drawn over it. Then result in interpretation. If you are going to write any sort of the interpretation, it will be after results only. So you can write it down later, show me in a record book. And if you are looking to write many things, you keep one more additional page. You can write it down left hand side, right hand side, protocol and observations you are going to draw and write in left hand side. So if you are looking to write it down some additional notes, so it will come after requirements. So you can show me at least 1 or 2 paragraphs are there. or afterward also, after interpretations, or you can write the additional notes. If you want to write any additional note, just show me. So it is already ready, this material. Okay, first experiment, second preparation of solid media. More or less is similar to first one. Then the four methods, strict plates and others are there. Yes, here I have kept that draw the diagram for each strict method on the right hand side by yourself that I haven't given. Okay, so what you need to do? You need to write it, draw the properly one round. You can use, sometimes people are having the ring. Otherwise, you can use the any additional things to make it the rounds at least, and you need to draw. The drawing part I haven't shown over here. How to draw? That is for the direct streaking sector wise. So whatever the number of the sectors you want to draw and that part, that you can, no problem. Already I have told you people in theory. Same thing is going to come over here. So drawing will be done on your left hand side page. For each strict pledge method on right hand side by yourself. Okay, sorry, it is please correct it as the left hand side, because you are having like journal is there. Okay, journal you are writing on the right hand side. Okay, and drawing is required to do on left hand side. Just correct it. Okay. હા એટલે ઇન્ટ્રોડક્શન્સ માં આવશે જ. ઇન્ટ્રોડક્શન્સ ને રિલેટેડ જ્યાં પણ ડાયાગ્રામ છે ઇન્ટ્રોડક્શન ની સામે ત્યાં કરશે. હા લેફ્ટ સાઈડ જ બધી જ વસ્તુ લેફ્ટ સાઈડ બ્લેન્ક પેજ ઉપર. Sometimes it may be there as the page will turn. Okay, it may be a left hand side, right hand side. So it is better as a blank page. Consider as a blank page. Sometimes some journals are there. It may be different. It is maybe not drawn properly. Otherwise, it is the initial page. It will be coming in left hand side. It may go in the right hand side. That is why I was opening one book journal. It was in right hand side, blank page. That is why it was in my impression. That is in right hand side. That's why I have written. So I don't know. Maybe at some place it may be varies from one to another. So on blank page, consider as a drawing the diagram on a blank page. Then the isolation by the spread plate method, it is going to remain same. So I have not written a lot thing. You can see. For plate method, draws the diagram, only 1 or 3 paragraph. I haven't given a lot loaded work. I have seen the others people's journal. Okay, that you are going to write and rewrite, nothing else. That burdensome I'm not looking to give you people. To the point, what is required, you may just write. Okay. Then effect of chemicals. Here it is going to, you can see more or less remains same. 2 paragraphs are coming for the pH, then for the temperature, dyes, everything. Introductory part, paragraph is going to remain same. Okay, so when you are going to write these things in exams also, whenever the practical session is there, you have to write these introductions, requirements, and other part is minimum required things. Okay, so it will be going to mug up otherwise. You are going to at least write this thing in a practical session because you have written so many times. Okay, so you are not going to forget it. If it is this part is going to ask. Then growth curve bacteria. Okay, it may be long to 1 or 2 page, not more. Then the qualitative analysis of milk. It is maximum 1 or 2 page. So in total, our full journal, just I have given is a 22 page. But if you are going to shrink it down, it may be maximum 12 page. Okay, nothing more. So you may able to start to write. I'm going to give the accordingly to dates. Okay, maybe next Monday, Tuesday. Today is a Monday. On a Thursday and a Friday, we are going to check the record books. Accordingly, you can start to write your journals also. Okay, and on a next day, Monday and Tuesday. So what will be the? Yes, we can finalize 11th. Okay, on 11th, you can submit to me. So I'm planning for your exams after the 20th. Okay, between 15 to 20th is there. So it may be between 18 to 23rd. It's starting from the 15th to 31st. We have to take. So here the slot time will be there. So I'm looking to take your exams before as much as possible. Otherwise, on a next week, last week, you are going to have the two papers together. And their journal writing may be going on. It will be not an easier task, right? It will be a not easier task to complete the practicals as well as to write it down. I don't know about the other papers situation. Mid-sem. Right. Yes. So as the mid-sem is going to complete, yes, and I think I have told these things earlier also, right? When your mid-sem one was there, one or two, one was there. I told that mid-sem two will be on this so many dates. You are going to have your exams as well as your practical journals may be going on. No, if I haven't told, I don't know about the other faculty, whether they have told or not. But according to me, if I'm not wrong, I have already told that it will be a very complex. Those who are regular into it, they may be knowing. So, and yes, as the practical session is going to work, you are going to have your some Diwali vacation. That's for it will be considered your reading time. And finally, you are going to have your comment. Yes, that's it. In next month, you can check your portal. Date will be there. It's somewhere it is in mid-November. Exactly, I don't recall the dates. It is mid-semester, mid of your November. So it may be about 10 or 15 days vacation is there. Diwali vacations or reading time you can consider. It may be, it's not a vacation, but you can consider the reading time. Because here it is the Diwali holidays will be there. Okay, about 10 days Diwali holidays there, and the final exam is next to it. Yes, final exam after Diwali. Wait. Huh? Yes, as the final exam over, it's your semester exam end, semester end. Huh? Ah, yeah, that part you just check it out and talk to the sections, science student sections below to it. જેટલું requirements લખી છે ને so just once again to correct એક વાર ધ્યાન રાખો by chance maybe some people has not understood or the query is there once again I'm telling okay so when you start to write so this part is starting either it's the left hand side right hand side where oh sorry one side I'm considering this one is a blank and this one is your lineate part. So you will start to write your first experiment number, title, aim. Sometimes may be a principle, sometimes start with the introduction. Or it may have introduction and principle together. Okay. Yeah, she is having that journals also. Very good. So enzyme kinetics or something is there. So you are going to start to write in this. So first part, it will be introduction and then you are going to have the materials or the requirements. In few only I have written the material. The simple reason is that something is totally new that you are not knowing. Okay, so in advance I have given this part. So I have written as a material. So requirements, till this it may coming here or it may go on the next page, no problem. Or sometimes some figures and are there related to introductions that you can draw on the blank page, whether it's the left hand side or right hand side. So it may happen that if you are going afterwards, okay, whether it is a page going up and side, blank side, you have to draw it. and you are going to then after write the protocols. Protocol always on a blank page. You are not going to write over here. Protocol you are going to draw or write. You can use your pen also. You can use pencil. Doesn't matter. Okay, while writing or drawing, you can use pen or you can use your pencil. But if you are using the pen for drawing, you may use only a black pen. Pencil or pen, you must use only a black pen, not a blue pen. Okay. હા આ સાઈડ જે કરો છો ને અહીંયા તમે પ્રોટોકોલ લખો છો, you can use the blue pen. Drawing, figure drawing, you can use the pencil or black pen. Okay, so you may pass these informations. I think you can do it to others. So otherwise everyone is going to call me. I can record their name. Okay, I will say everything. Many a times they can call. Oh sir, I was absent on that day. I will say no problem. Okay, I will say no problem. Okay. So I'm going to keep their track records. They all were absent-minded and they haven't taken the instructions properly. Okay. I will never mind to repeat the things again and again. Okay, I'm telling, but after completing and telling so many times, if the person is repeating, that means there is only two things. Either one is the one fully absent-minded, never been in a class or never been looking or present but remain absent. And another is they are just looking to make a persons in some trouble for him and her and as well as to the below to before them. They looking or would like to become a somewhat smarter. Okay, so I will be not going to be over smart. I will be as general as such. Okay, I'm going to just note it down. Okay, here, what is your number first? Tell me. What are the instruction is required? I will repeat. Okay, so here the observations and results. Results you are going to write on your line 8 part only. Okay, and if any interpretations or is there, you can write it down below to the results. Or something is there that if you want to write, you can write it down below to the observation table also. That is up to you. Sometimes space is there here and there. Okay, you can write it down either of the place. તમને લખાવી દીધા ને મેં રિક્વાયરમેન્ટ્સ નહીં, રિક્વાયરમેન્ટ્સ તમારી જે છે ને એ લાઈન ની નીચે ઇન્ટ્રોડક્શન્સ ની નીચે આવશે. આ સાઈડ પ્રોટોકોલ જ ઓબ્ઝર્વેશન્સ અને પ્રોટોકોલ બે જ વસ્તુ મેં તમને કીધી છે. પછી કઈક એડિશનલ નોટ્સ હોય તમારી, બરોબર છે? ઇટ ઇઝ બેટર યુ મે રાઇટ ઇટ ડાઉન ઓર ઇન્ટરપ્રિટેશન બિલો ટુ ધ રિઝલ્ટ. ઓલું બોક્સ હા હા એટલે હું રેકોર્ડ બુક્સ કહું છું ને, રેકોર્ડ બુક્સ કહું છું એટલે તમે શું કરીને લઈ આવશો એટલે તમે પછી ભૂલ થતી હોય તો હું તમને કરેક્ટ કરી શકું. You can use pencil, blue pen, black pen, no problem. Drawing is must pencil and pen and only a black pen. હા ઓબ્ઝર્વેશન પ્રોટોકોલ. હા ડાયાગ્રામ ઓબ્ઝર્વેશન્સ ની અંદર જ આવી જાય અને ડાયાગ્રામ કદાચ અલગ હશે તો પણ એ આપણે અહીંયા જ કરશું. યસ, બધું રાઈટ હેન્ડ સાઈડ. હા હા એ તો લાઈન વાળી જશે. So now I hope it's clear. Huh? Right hand side. Yes. Yes. So it is always advisable because if you are going to write something, interpretation or additional notes is there, right? So if you are writing, sometimes the people are writing to start here, but going in some different patterns. So whatever the additional thing is there, additional notes or the interpretations, you may write on the line 8 part only. Okay, otherwise may happen that some people may start below to it, but when they will started to write like this, but then when this is going in upper part. Okay, it is a blank page. Okay, so it is written like started here and going here or sometime below. It seems like a mountain, so don't write. Better only protocol, figures and observations on a blank page. Everything else, including additional notes and the interpretations, may write on your line-aid page only. Okay. If anybody wants any clarifications, you can talk to Beam also. Okay, I hope he has understood. Sometimes those who are internationals also, they may have some query. They can talk with him. If not solved from Beam also, you can come to me. I'm always ready. Okay, before a day of a practical also, I will be ready. But then don't expect how to perform, what to perform. Yes, and on exam day, every individual must have their own matchbox, then their marker, apron, and microbial. Every individual must have their apron, microbial, matchbox, and their marker. Anything in my practical sessions, even when I'm in invigilations, anything, just change of a ball pen, pencil, check rubber, I consider as a material transfer between two. You know the material transfer means? The copy case. Okay. Just change of any sort of the things, even a marker or a ball pen or a pencil between the two, I will consider as a material transfer. Yes. So if you are coming without matchbox, you cannot perform, I think, certain experiments. Okay. So requirement will be there. You can sit idle properly. When your time is completed, 10 or 15 minutes may be given to you to perform at one burner. Okay. So you may sit, your time is get over, you may go. I will not allow to take the things from your partners or colleagues. It is a compulsory, it's a practical test. Okay. It's not a regular days. So don't expect leverage from my side. Yes, I can give. Okay. But it will be, I will consider as a not perform. If you are not having the by chance matchbox, okay, because it is your exam, but maybe by chance it's forgotten. Okay, you can perform, but I will keep that this part was not there and it will be considered from my end as a not perform. Okay, it means you have done, but nothing is done. Okay, on more request I may allow, but you will be just considered as a why? Those who have taken it, they have put their efforts to bring this material. How I will give the injustice? Because they have done it, they have brought, and those who haven't brought, just based on the one request, they can do it. So it is not permitted. Okay. And I have told in advance. Okay. It is not also that I'm telling a day before or the just right now it's and then your practical is there. It is not also in that hurry. So now practical sessions related work is, I think it's complete. Okay, so let me continue for our that topic. આપણે મારે તો આપણે જો કઈ કરવું જ નથી, આપણે એક્ઝામ લેવાની છે. એક્ઝામમાં માણસ પાસ થઈને ફેલ થાય. તો જીવન એક એક્ઝામ છે, એમાં શું કરવું? નહીં. Yes, it is competitive world. It is really, it's competitive world. Always you can do it. પછી બધાને એમ જ જઈને ન કહેતી કે ભાઈ કહેતા નહીં કોઈને, પછી બધાને કહી દે. Okay, okay, let's move ahead. હા સિમ્પલ સિમ્પલ, એમાં કોઈ હું વાઈફાઈ ખર્ચા નથી કરાવવા. Not required introduction, no need to write introductions in record book. Olama બધું. Okay, 5 minutes is there, 5 or 10 minutes. Let me complete this topic also. Next part, how we can measure metabolic growth is there or not. Growth can be measured based on the metabolic activity. That if something is growing, by chance is in a lag phase or in stationary phase, you can observe very easily. Initially, maybe a color change is going to happen. કોઈ વસ્તુ બની જાય બરોબર છે. So when it is cooked, we can based on the observations also we can see. Or sometimes it is a solid material. Either it is a liquid or solid. You may have kept something on growing on a plate. So on a next day, you may see the growth. So just by observation also, we can understand there is a growth. Here it is like some bowls or the glass is kept. So something you may inoculate. On next day, you may observe there is a bubbling formation, is the fermentation. Okay, the fermentations can be observed. So metabolically, certain things may start. Yellow color, green color, color change may happen. Okay, that is a product formation. And these product formations can be measured visually also. Or you can measure even. That can be measured based on the assessment. Each metabolic process having their different. And the last point is related to dry weight. Dry weight is the last ultimate point over it. What we can do is how we can get the bacteria simple. Yes, he is playing very well. Let me take. If I want to see MPN method, okay. MPN method, other method is there. એટલું દોરી નાખ્યું. ના ના હવે હું રમું ને ઘડીક વાર તમે રમી લીધો હું રમું ને. So how we can get the dry weight? For the dry weight, how we can do a collection of the cells? One is the filtrations that already I have talked. Other is a centrifugation that I can keep the whole tube. Otherwise, I may distribute them into small centrifuge or in a bottle. And whole bottle we can do a centrifugation. So cells within them is going to get pelleted at a bottom. Okay, so within one simple procedures, all the cells can be collected. If we are in a lag phase to log phase or stationary phase is there, maybe after 2 or 3 days, you may see that is a number of the cells. The turbidity is very high. And I want to get all the cells to be measured. So what I can do, just do a pellet formation. Pellet it out and just make it dry. How we can dry it? Keep the tubes into hot air oven. Within 24, everything is going to get dried and powder will be there. Just measure that powder. Okay, just measuring the powder, we can say there is a bacterial growth is there or not. Sometimes we are looking for bacteria as well as the fungi. Major thing is related to fungi because they are having some mycelia formation. Then mycelial formations you can collect into one tube at the bottom. And this part you can then after once again reuse for your purpose, or sometimes it is being sold as a product, like it's the yeast powder. Baker's yeast ma jaiye na, Baker's yeast is coming as one tubes or the powder only. So what they have done, simply they have inoculated one flask for a given time period, and then after they centrifuge it. So as the centrifugation, the liquid material remain at the upper part, super layer, okay, and supernatant is called, and below its pellet formation is there. The supernatant is removed, just tilt it down. No, okay, tilt it down, and supernatant is gone, and then dry it. Dye powder is packed into one tubes or in a box and sent to for a selling. So we never open our yeast powder in a sterile conditions generally, and we are keeping in a freeze. So it is already dried. It required medium to the grow. So here there is no problem. So we are going to measure bacterial growth or the microbial growth based on their dry weight. So here it's the last point related to dry weight. So it's a very and convenient process to perform. People are performing based on their easy methods. Okay, so I hope this point is clear to everyone. Okay, so only 3 methods we have gone here: turbidity, metabolic activity, and the dry weight. Okay, these are the indirect method and direct method we have gone through. They are plate count, pour plate and spread plate. Already we have done. Serial dilutions you are knowing. Filtrations just I have told. MPN and microscopic count. Today just we have talked about microscopic. Earlier we have gone for the MPN. There is a 5 tube practical in 3 set. Okay, maybe you do remember. That is minimal probable number that we have seen. So this all question direct method can be asked. Sorry, can be asked into 8 marks, 6 mark, or any individual method can be asked into 4 marks to you. So I hope it's clear. Okay. So put your attendance. Accordingly.